Hey everyone, welcome to Bridgeside Point 2, one of 4 research buildings used by the PMI program. My name's Sydney, I'm a 4th year, and I'm going to give you guys a tour today, so come on. Welcome to the building. So this is Bridgeside Point 2, also known as BSP2. And so even though Pitt has several different research buildings, all of them are self-contained. So here on the first floor is our animal facilities, and up on the 4th and 5th floor are where the MMG department labs are. So I'll take you to I'll show you those now. Okay, so welcome to the 4th floor. Like I said before, the MMG department's on the 4th and 5th floors, but they look exactly the same, so I'm just going to show you the 4th floor today. So on this side of the floor, we have like all of the offices and break rooms, and then to this side over here, we have the labs. So I'm going to show you guys to the labs. So there are 6 labs on the 4th floor, and we're going to take you into the Cooper lab so that you can meet one of their lab mates, and they're going to show you around their lab and also tell you a bit about their research. So come on. Hey, Shelly. Hey. Hey, so we're doing a tour for the interviewees. So we're wondering if you could show us around your lab and tell us a bit about the Cooper Lab's work.
Sure. So, as Sid said, I'm Shelly, and I'm a 5th year grad student in Von Cooper's lab, which is where we're now standing. So my lab studies microbial evolution first and foremost. And generally how that shakes out is that we take bacterial populations and we expose them to selective pressures, something like antibiotic pressure, biofilm lifestyle, nutrient adaptation, really anything that we think is scientifically interesting and clinically or environmentally relevant. And so we expose them to those pressures and we allow them to evolve and adapt within those environments. And then we use whole genome sequencing of bacterial populations to identify the mutations that we're selected for and further study evolution in those environments. So today I'm going to show you around my lab and show you some of the equipment that we use to do that work. First of all, so as Sid said, we're in BSP2, and this is pretty much what all BSP2 labs look like, but with a different view. We're lucky to have nice big windows, and we have bench space and reading and writing space for each of us. This is the expensive room within my lab. So this right here is the sequencer that we use for bacterial genome sequencing. It's an Illumina NextSeq 500. We're lucky enough that we can do a lot of our sequencing in-house, but there's other areas and centers on campus that also do sequencing and have those sorts of capabilities and resources. We also have this liquid handling robot and DNA extraction cube. So lots of fun gadgets that we get to play with. And when we're done doing our experiments, we generally come out here and we can write up our work, go see other grad students on the floor. We have a nice view of downtown that you can see here. So you can see the US Steel building, which is the one that says UPS PMC on it. And across the river, you can get a view of Southside. See you later.
Yep. Okay, so come on guys, I'll show you some more of the stuff on the 4th floor. So microscopy is a huge component of many of the lab's research on this floor, and so we're going to show you guys a couple of our cool scopes. So we're going to start with this one. Let's see if anyone's in here. Oh look, it's Jamie!
Hi, Sid.
Hey! So we're giving a tour to the interviewees of the building. We're wondering if you could tell us a bit about your lab's work, whatever this cool contraption is, and what you do.
Okay. Hi guys, my name is Janie French. I am a 2nd year student in Seema Lacta-Wallace lab. This microscope, we call it our dye spin. It is a dual light sheet microscope, and it was actually assembled by a former postdoc in the lab. Right now we use it primarily to do live cell imaging, especially with cells that are infected with influenza that have a fluorescent reporter included in them to help visualize cell infection. So that's just kind of like a broad overview of the microscope, but it's pretty special. There's not a lot of them out there, so it's a really cool tool that we have.
Can you tell us a bit about your work?
Yeah, I am working on a transmission project. For those of you who don't know, Seema's lab particularly specializes in influenza virus assembly, but also transmission. So my project looks at how influenza co-infection with Streptococcus pneumoniae impacts flu transmission. So right now I'm doing some co-infection work and trying to develop an in vitro model for that. Yeah.
Super cool. Okay, and then last question. Can you tell us why you chose PMI?
Yeah, so I chose PMI for a couple reasons. One thing that was really close to my heart was obviously as a female scientist, I wanted to work in an environment that both had a lot of women and had a lot of female PIs. And this was one of the only universities where I saw that there was a lot of representation, and it seemed like the university supported female scientists. And so that was really cool. That was one reason. I also loved all the grad students I met on recruiting weekend, so that was a huge factor. And I just really enjoyed all of the PIs that I met. They all seemed really down to earth and relatable. So those are some of the reasons that I chose PMI.
Yay, thank you so much, Janie. So we will leave you to your work. But yeah, thanks.
Yeah, thank
Bye.
you. Bye, guys.
So, like I said, a lot of labs on the floor and in the building use microscopy, and so we're going to show you the departmental scope room. So, like I said, lots of scopes. So here we have our 2 departmental confocal microscopes, and this one here, the Nikon, is brand spanking new. So very excited to use that one. And then some labs also have their own personal scopes. And oh, it looks like Allison is currently using the scope. So Allison, we're doing a tour for the interviewees since they can't come, and so we were wondering if you could tell us about yourself and your lab, and yeah.
Hi guys, well like Sid said, I'm Allison. I'm a 4th year grad student in Jen Baumberger's lab, and I study interbacterial competition and interactions and see how bacteria communities change during things like viral co-infections. So I use the microscope to look at how bacterial communities form, and so like here you can see these green bacteria or biofilms. So we can use these live cell systems to actually watch bacteria interact with each other and form these communities.
Very cool. And can you tell us why you picked PMI?
So I chose PMI just because the people here are so friendly and so collaborative. It's so easy to walk across the hall and talk to another professor about your research and get their ideas. And so it's just a very fun and easy place to be.
Yay! Okay, thanks so much, Allison. So, yeah, guess we'll keep going with the tour. Oh, yeah. So this side of the floor is more of our like office space. So this is where we have conference rooms, the PIs have their office, and then there's also cubicles for the postdocs. So with work from home, this side of the floor has been pretty empty lately, especially on a Friday afternoon, which it is today. But we can look around and see if anyone's here to say hi. So come on. So these are the offices. These are the postdoc cubicles. And oh look, a light's on. Let's see if anyone's here. Oh look, it's Paola. Hi Paola, we are recording a video for the interviewees. Say hello.
Hi everyone.
Okay, let's go. So this room is our classroom, and so if you guys were here interviewing in person, this is where you get all of your presentations from. And this is also where obviously you would have your classes. It's where you would give your research and progress every year if you're in the MMG department. This is where we also have our weekly MMG seminars where we have PIs and other speakers from around the country and around the world come and give weekly seminars. But yeah, let's see if anyone is in here. Oh look, it's Allie. Hi Allie. We are giving a tour to the interviewees since they can't come. So we were wondering if you could tell them a bit about yourself and your science.
Yeah, sure. Hi everybody, so I'm Allie. I'm a 2nd year student at PMI and I'm in Jen Baumberger's lab. Currently I'm going through the comps process right now, which is why I'm here in this room studying at the whiteboard. And so whenever you're a student here, your comps process will take place around your 2nd year after you've chosen your lab and gone through the rotations in your 1st year. And so comps is just a process that we go through that is a 2-part process. So it's one part of a written exam where you'll write on a proposal on the topic that you want to cover during your thesis here. And then once that gets approved, you will do an oral presentation on that, and then your committee members and that will ask you questions about your project and any other sort of general knowledge. And so it's a big studying part of the year, and then you get to finish that up and then you get to start your research.
Yeah.
Very cool. It's a very stressful time, but Allie's going to do awesome. So could you tell us also why you chose PMI?
So I chose PMI because I was already actually an undergraduate here at Pitt beforehand, and I really loved all of the collaborative environment and all of the great research that was being done at Pitt. And so I started in the biological sciences department, and then I chose PMI because I was really interested in microbiology and immunology, and this was the best place to look for that kind of stuff.
Perfect. Well, we will let you get back to studying, Allie. So come on, guys. I'll show you the break room. So the last stop on our tour is the break room. So it doesn't get too much use now during COVID, but maybe, oh look, it's Grace. Grace can tell us a bit more about the break room. Hi Grace, so we're doing a tour for the interviewees since they can't come to Pitt. So we were wondering if you could introduce ourselves and just tell us about like the break room and how things used to be before COVID.
Okay, so hi everybody, my name is Grace Carreno Flores. I'm a third year student at Bomberger Lab. And well, this is our break room. I think that it is a quite big space and comfortable. So before COVID, we used to do like baking sales and happy hours. But I promise that it will be back. And now, what I like the most about this space is this, where we can see the river and south side. So as you can see also, the bridge that shows that this is close to south side, so it's at a walkable distance. So yeah, this is pretty much a good space for me.
Yeah.
Like to have lunch or make yourself a cup of coffee. So yeah.
Yeah. And then can you tell us why you chose PMI?
I chose this program because I think that we have like really exciting research in here, and like I like the host microbe interaction. I found like really good research about that here.
Yay! Well, thank you so much, Grace, and thank you guys for tuning in to our tour of Bridgeside Point 2. I know you guys can't be here, but I hope this tour was super helpful for you guys seeing our research space and give you at least a little hint of what it'll be like to be a grad student here at Pitt. So good luck with your decision, and I hope to see you here in the fall. Bye!